when Earth originated around 4 billion years ago, that's the time we believe they originated. Okay, and how do we know that? We know that by the presence of the fossils that were formed by them. They are very, very small, the prokaryotes, and they fall into two major domains. One is called bacteria and the other is called archaea. Now, bacteria are the early cyanobacteria. Remember cyanobacteria? They used to be known as blue-green algae until it was found that they contain a prokaryotic cell. That's when scientists started calling them cyanobacteria. What does cyano word means? Cyano means anything. It comes from cyan, meaning blue, but of course we have the green component also, because they have green as well as blue pigments in their cells, and because of that they undergo photosynthesis. Okay, and when some organism performs photosynthesis, you know what is the end product of photosynthesis? It's oxygen. So because of that, the early cyanobacteria produced oxygen, which actually allowed for the rest of the life forms. Okay, and that's how other life forms were introduced on Earth. Okay, now let's talk a little bit about archaea. What are archaea? Archaea are the earliest life forms, prokaryotic of course, but they are extremophiles. Why they are called extremophiles? Because they can handle extreme conditions that were present in the early history of Earth. Okay, means even now you can find archaea. If you remember, I said they can be found in the hot geysers of the Yellowstone National Park. Okay, they can live under temperatures that can range from 110 to 130 or 50 degrees Celsius. So they can handle as high of a temperature. They can grow in the deep sea hydrothermal vents. They can handle that high of a pressure. Okay, you know how much high pressure is there inside the oceans. They can handle that also. They can live in extreme cold also in the polar ice caps, Antarctica regions like that. So all these things, because of these extreme habitats that they are used to, they are called extremophiles. Okay. Now, archaea and bacteria, out of the three domains of life, just these two belong to the prokaryotes. And what is a prokaryote? As I have explained on this board, this is a simple prokaryotic cell. Prokaryotes means they were born before the nucleus was discovered. Okay, so that is a prokaryote, and their cell is very, very simple. All they have is a cell wall. Okay, cell wall, and they have a cell membrane and a DNA in the center of the cell. The center of the cell is also called nucleoid. It's not a nucleus. So there is a difference between nucleus and a nucleoid. So pay attention to that. Okay. And the DNA is found in them in the nucleoid region, which is in the center of the cell. And the other thing, the dot-like structures that you're seeing, they are just ribosomes. And apart from that, they have little hula hoops little circular pieces of DNA that are called plasmids. Okay, the two prokaryotic domains, archaea and bacteria, are not closely related, as you can tell from that cladogram. Okay, as you can see, domain bacteria is slightly uh, on a different branch than that of the domain archaea. If we look at this cladogram, we get the characteristics of the prokaryotes. So in prokaryotes, like we said, we have unicellularity. When we say unicellular, uni means single, right? Means they are single celled organisms, and they may, what I said is, they would like to stick together to each other and will either form a colony or they can form biofilms. Okay, so in case of bacteria, we use this term often because biofilm doesn't mean that they are grouped together to each other. In biofilms, they are also attached to the surface, not just to each other but also to the surface. And a very easy example we can of biofilms we can find in our mouth. So as you know, we have some uh, like people, those who have dental caries. Why do they have it? Because they have that tartar, layer of tartar that forms on their teeth, right? What is that? So here the groups or the colonies of those bacterial cell are sticking not just to each other, but they are also sticking to the surface of the teeth, and that's what makes a biofilm. Okay, now their cell size is ranging from like less than you know uh, a micrometer to slightly more than a micrometer, but then usually they are considered in size less than micrometer in size. And if micro is something that you guys are wondering what a micro is, my amount of the micros measurement is 10 to the negative 6. That means you need 6 zeros, meaning this means this 1, 2, 3, 4, 5, 6,000. 1 to the negative 6 is what makes a micro. So now you can see how small these microbes are, these bacteria and archaea are. Okay, that's why, when remember in the lab I told you you need a 100x objective in order to view them well, okay, which is an oil immersion lens. Now when we look at their chromosomes, they have circular chromosome in the nucleoid region. This is the nucleoid region. The region that is in the center is the nucleoid region, and there is just one single circular bacterial chromosome, of course. And chromosomes are made up of DNA, so you can also call it a DNA, right? And apart from that, they have some another type of DNA which are circular as well, and they can be located in any part of the bacterial cell, and they are called plasmids. Okay, these plasmids are responsible for very specific genes on them. A couple of example includes the gene for antibiotic resistance. Okay, so it is the plasmids that have the ability to jump from one cell to another, and that's why I have given them a little nickname called hula hoops, which kind of like feels to me like they jump around and spread their gene in the bacterial population. Okay, now cell division. What type of cell division do these um, prokaryotes undergo? So they have mostly asexual reproduction that is called binary fission. Okay, so what happens in binary fission? In binary fission, a cell divides asexually and it divides into two. So if this is a bacterial cell, okay, it just simply is going to duplicate its DNA and it's going to divide by a septa, and from one cell you see two cells are formed. Okay, there is no mm, complex cell division process like you are aware from your previous classes, how you have cell divisions like mitosis, meiosis, where you have different kind of stages, acronym being PMAT. Okay, here we don't have anything like that. It's just one cell duplicates its DNA and then divides it into two. So that way both the daughter cells, they get their DNA. Now, in some cases, we do see some sort of genetic recombination. When we said recombination, it means that this here, these bacteria can undergo horizontal gene transfer. So, what is horizontal gene transfer? Horizontal gene transfer would be like horizontal gene transfer is when the genetic information goes from one cell to the other cell in the same generation. Okay, this is not a nucleus. Okay, so I'm not, please don't misinterpret it as a nucleus, because prokaryotes do not have a nucleus. Okay, this is just the DNA. Okay, so what happens is this is called horizontal gene transfer. What happens here is, what is vertical? Let me first tell you about vertical. Vertical gene transfer would be when it undergoes vertically from a parent to the offspring. Okay, and where did the parents get it from one generation to another will be the vertical gene transfer. Okay, now horizontal is in the same generation, but there is genetic recombination. What does that mean? Means there is some sort of swapping of the genes or transferring of the gene that's occurring. We know in vertical, like mitosis, meiosis, as you know, we get the transfer of the genetic information, right? But here, this is the bacteria or the prokaryotes way to exchange some of the genes. Okay, so that the offspring that's formed is not exactly similar, because that occurs only in asexual reproduction like binary fission. Okay, now if we say horizontal gene transfer, this can be introduced by three ways. Okay, I think we will be studying this in the next subsequent slides, but for now you can think of transformation, that's one type, transformation, conjugation, and transduction. These are the three types of horizontal gene transfer. Okay, so another thing that they have in them is the flagella. So flagella is actually a single fiber of protein, and it doesn't have to be a single, like necessarily. In bacteria you have different types of bacteria, different types of flagella, and they usually act like propellers. Okay, so the different types of flagella would also, we'll be studying in more details. Now, in, like I said, as we talked about their bacterial cell structure, they don't have too much compartmentalization, meaning they don't have any specific type of organelles like mitochondria or endoplasmic reticulum or lysosomes and things like that. have structures that can resemble like organelles, but it's not really an organelle because they don't really have, you know, uh, the membrane around them. Now, plasma membrane can be invaginated. Okay, so how do you think like a cyanobacteria? How does it have? It doesn't have a chloroplast, right? But what does it have? The plasma membrane gets invaginated, and that's what it isolates. You know, those um, thylakoids or the chloroplast containing structures in like a separate chamber. Okay, but it's not really a phospholipid bilayer membrane, which actually would be an envelope. Make sense? Or they can isolate a certain area inside the cell, and you know there will that will be responsible for respiration. So it's not really going to be a mitochondria, but it's something in that area that will designate it as to conduct respiration. Okay, and they are metabolically very diverse. They have oxygenic and oxygenic photosynthesis. Okay, and they are chemolithotropic, means they will use some types of chemicals in order to conduct their metabolism. Now, archaea and bacteria both lack a membrane bound nucleus and also an endomembrane system, but they are distinctly different cell types. These are the major differences between the archaea and the bacteria that we should understand. So, in archaea, the plasma membrane or the is made up of non-polar hydrocarbons called isoprenoids that are joined to glycerol phosphate with ether linkages. Whereas in bacteria, the fatty acid chains are joined to glycerol phosphate with ester linkages. This is the one that we know uh, is present in the eukaryotes also. So in bacteria, the fatty acids are joined with ester linkages, whereas in archaea, it is joined with ether linkages. So that's how the archaeal plasma membrane differs from the bacterial plasma membrane. And also, archaea have glycerol 1 phosphate, whereas in case of bacteria, the stereoisomer of glycerol 3 phosphate is present. In case of the lipids that are found in the archaeal membranes, those lipids can be branched, they can be cyclic in shape, they can be made up of tetraethers, and they are capable of forming monolayers. But in case of bacteria, these membrane lipids are always unbranched, and they will form a phospholipid bilayer only. Okay, so these are the major differences between the membranes of bacteria and archaea, and that's why they were promoted to a separate domain. These are the examples of those pictures of those membrane lipids. So chemically, archaeal plasma membrane are different from the bacterial plasma membranes. Now, other differences, that archaeal cell wall does not have peptidoglycan, whereas bacterial cell wall is made up of peptidoglycan. Okay, what is peptidoglycan? Peptidoglycan means they are glycan means sugar, peptido means protein. Okay, so what happens is that our in bacteria, the cell wall is always going to be made up of peptidoglycan. Peptidoglycan is this peptidoglycan. Glycan is sugar, it represents the sugar component, whereas peptido represents the protein component. You know, amino acids are joined by peptide bonds in order to form a protein, right? So that's where the peptido is coming from. In case of archaea, they have some other material called pseudomorine that makes them. And the S layer outside of wall or replacing the wall. So they in archaea, they are composed of glycoproteins that form a rigid paracrystalline surface. The flagella of archaea are very unique and they are evolved independently than that of the bacterial flagella. So that is also another difference, unique archaeal flagella. Other thing is the DNA replication of the archaea. They both have single replication origin. Okay, the archaeal initiation proteins are similar to the eukaryotes. Okay, initiation proteins, the proteins that initiate the DNA replication are very similar to that of eukaryotes. Okay, and archaeal gene expression is also similar to the eukaryotes. If you remember, they both have transcription and translation coupling. Okay, and so in overall their machinery, okay, is similar to that of eukaryotes. They also have histone proteins just like the eukaryotic proteins. Okay, now archaea have undergo methanogenesis. What does that mean? They are going to make methane gas. So they are all organically produced uh, methane that exists comes from these archaeal cells. They also carry out the oxidation of ammonia, anaerobic ammonia oxidation to nitrogen gas is only found in archaeal kingdom, in archaeal domain, I mean. Okay, and they are the major contributors to the global nitrogen cycle. Okay, the nitrogen cycle that you've studied in your ecology, it comes because of through archaea. Now, what are the shapes of the bacterial cells? So the three different shapes of the bacterial cell is first is cocci. Cocci means they are circular bacteria, circular or round. Okay, you can memorize them like that. Then second shape is bacilli. Bacilli means bar-like or brick-like. Okay. Also, they are called rods because they look like this. Okay, circular or like these rounds. Then you have spirali. What are spirali? Spirali are like small spirals. This is how you can memorize them. Spirals, small, like this, like this, like this. Okay, small snakes or spirals. They can look like this, like that, helical shaped. Now, cell wall of bacteria is made up of this chemical called peptidoglycan. These are very, very important for the bacteria in order to maintain the shape, and they are always able to withstand hypnotic environments. Remember, in when you have hypotonic, not hypno but hypotonic situations, what happens? The water enters inside the cell, right? And if there was no cell wall, okay, around the bacterial cell, then what would happen? Because they are, yes, because they are always present in a like a um, fluidy environment, so water would very easily enter inside them, and because of osmosis, yes, it will lyse the bacterial cell. So that's why peptidoglycan cell wall is very, very sturdy, and it's a rigid network that gives them that maintains their shape and does not make them prone to cell lysis. Okay. Now, how do we differentiate bacteria into two types is all dependent on this cell wall. Okay, so because of the cell wall presence, the presence of peptidoglycan cell wall in a certain way, that bacteria have been divided into two main categories based on the gram stain. Okay, so bacteria can either be gram positive or they can be gram negative. Okay, so when we say gram positive, then that means gram negative. What does that mean? When we perform gram staining, we use four different types of stains. Okay, the first stain that we used is called crystal violet. That's the primary stain. First stain, primary stain. CV stands for crystal violet. The second stain, we use a mordant called iodine. Okay, mordant. The third chemical that we use is called acetone alcohol or just alcohol. Just depends what your lab has. That's what we will use. The fourth is this acetone alcohol, I'm sorry, is a decolorizer. Okay. And fourth is a counter stain called safranin. It's a dye just like crystal violet, but it is used as a counter stain. Okay. So each one of this chemical, as you can see, has a certain role to play in the gram staining process. So like I said, gram positive and gram negative have these differences in their peptidoglycan cell wall, and based on that, they stain differently in the gram staining process. Okay, so if you when you stain this is the procedure we follow. It's summarized in this picture, and gram positive bacteria have a thick layer of peptidoglycan cell wall. Okay, whereas gram negative have a thin layer of gram negative bacteria. Gram negative have a thin peptidoglycan cell wall. Okay, so what happens here is they also have one additional thing. If we are looking at this being a gram negative bacteria, then we have to understand that they have a thin peptidoglycan cell wall. Apart from that, they have an outer phospholipid bilayer which is made up of LPS, lipopolysaccharide. This will be their cell wall made up of peptidoglycan. So this is thin. This is made up of peptidoglycan cell wall. Okay. So cell wall is made up of peptidoglycan, but the outer wall is made up of LPS. If you look at gram positive, they will be having a very, very thick cell wall, peptido. So you see all this is thick. See how thick it is? So this is thick peptidoglycan. Okay, so this is your gram positive, this is your gram negative. Got it? So now what happens is, because of these differences in their cell wall uh, thickness and the presence of that outer layer in the gram negative, we face all these differences uh, while staining them with gram stain. Okay, so when you use the primary stain, what happens is, at this stage, both the cells, if you get a chance to observe them under the microscope, you will see that they have a different they both are going to have the purple stain. So at this stage, both of them will look purple or violet, because that's how the crystal violet color is. Both are going to be purple. But when you stain it with the iodine, what happens when you're staining with the iodine, which is the second picture you see, they will iodine acts as a mordant and it fixes the crystal violet stain inside the peptidoglycan cell wall. Make sense? So both at this stage also will look purple. But in the third step, you are going to use some, what do you call, a decolorizer, which will be alcohol. So why is it called a decolorizer? Because when you use alcohol in gram negative, remember you have a outer LPS layer. Okay, let me draw it with a slightly different color so that you can see what I'm talking about. So this outer layer is, when you use alcohol, this layer is going to, because it's made up of lipid, so lipid is going to break down this cell wall, outer cell wall. Make sense? It breaks down. So when it breaks down, that what happens, the crystal violet color is going to seep out from this. Make sense? In all the directions. Make sense? So the violet color is going to seep out. This means what? These cells at this stage are going to become colorless, because if there is no stain, the bacterial cells look colorless. But in gram positive, because there is no outer wall, you see that, so the effect of alcohol will be negligible, there will be no effect, and they will still retain their, they will still be looking purple, because rather it is fixed by the mordant iodine, so they will still be purple here at this step. Make sense? They are still going to be purple. Make sense? Now, because the gram negatives are looking colorless at this point, and you know, in order to see well under the microscope, we need some kind of contrast, right? That's one of the rules that we studied in uh, slides, in viewing the mic under the microscope, right? Viewing the specimens under the microscope, right? So if this is colorless, we need to have a counter stain. Okay, so we will apply safranin. Safranin is a reddish pinkish dye. Okay, so when you stain something that is colorless, it is going to look reddish or pink in color. But here we don't have that problem, because these cells are not, they are still purple and they are going to stay purple till the end. Make sense? So that's why the presence of what? Purple color all throughout the gram positive indicates that they have retained the gram reagents, okay, which is your crystal violet and iodine. That's why they are gram positive. Whereas the gram negative bacteria, they lost the gram color of the gram stain, and that's why they are called gram negative. And they, instead of purple, the gram negative look red and or pinkish in color under the microscope. Okay, and that's how you can tell from this picture. The purple colored cells, purplish or bluish color, they are all gram positive, whereas the pinkish cells, they are all gram negative. Got it? This is in nutshell the appearance of the gram positive and negative bacteria. Now this picture shows you the structure of the gram positive and gram negative cell wall. Okay, so here you can see here, this is the pink colored layer is the peptidoglycan layer. See how thick it is in the gram positive compared to this layer, which is so much thinner compared to this. From here to here, you have peptidoglycan, and this much, only this much. And also, there is no outer wall here, right? But here you have this lipopolysaccharide outer wall. This is lipopolysaccharide outer wall. So see, these are the differences as you can see under the microscope. Now, another other type of structures that can be found in the bacteria are like a capsule. What is a capsule? Capsule is like a gelatinous layer that is found around some bacteria, not all. Okay. So what happens here is that that gelatinous layer surrounds the cell wall. Okay. And it will look like, it helps in the attachment process. Okay, so it helps the host, it helps the bacteria to attach to the host. It also protects the bacteria from the immune system of the host, meaning they are more virulent now because they have capsule, means they are like, I kind of like to think of them like, they are wearing a hoodie. So when a person, think about a robber trying to rob, and he will wear a hoodie, he can easily sneak in and can, uh, in a store and can, uh, do what he needs to. So this is what they do. They can easily sneak in under that cover because it's an outer layer. They are like wearing a hoodie, and the host immune system cannot recognize them because they can't see through the hoodie, through the capsule, and they let them come in inside there, and that when they go there, they are there to cause the damage. Got it? Now, what are flagella? Their bacteria has flagella, like we spoke about earlier. They are like, they are made up of a protein called flagellin. Okay, and they are fiber-like, kind of hair-like, but they are much bigger than a. And then they are slender. They are rigid. They help in the movement of the bacteria. Okay, just like flagella, you have pili. Pili are much short structures compared to the flagella. Okay, now pili also aid in attachment. They are not meant for movement, but they help in the attachment. Okay, so if we are talking about, this is a bacterial cell and this is another bacterial cell. Okay, so a pilus is a small hair-like structure which will help attach it to the next cell. Okay, and they help in the process called conjugation, which is a kind of horizontal gene transfer conjugation. So these are short, much short. Flagella can be very long and hair-like, but pili are very small. Okay, so this is a flagellar structure of a gram-negative bacterium. So as you can see, they have three main parts to it. They have a base, okay. This is a base which is kind of anchored into the cell wall of the bacteria. Okay, if this is the cell wall, this is the cell wall, so it will be anchored inside the cell wall. Then they have a hook, so base, hook, and then the portion that is outside is called filament. Okay, so the filament is the hook, actually along with the base is embedded inside the wall. Okay, hook, this is hook. Got it? If this is cell wall, so this whole structure, uh, the base has an outer ring, protein ring, and an inner protein ring, and the hook basically helps it turn in the direction where it needs to go. Now, apart from that, bacteria have very unique structures which are called endospores. Okay, so what are endospores? Endospores are, they are also called just spores, but I like to call them endospores. Okay, some places you'll see they are just saying spores, bacterial spores. Okay, endospores are formed by some bacteria. Actually, there are only two genera in bacterial kingdom that can form these endospores, and their names are bacillus and clostridium. Okay, so these endospores are formed as a result of stressed out conditions. Stressed out conditions meaning they face a lot of environmental stress. Let's say there is um, dehydrating conditions, right? Because bacteria likes to grow in, a lot of, um, water, or if there is, or there is radiation, very high radiation. So to save themselves from desiccation, okay, some bacteria, they are capable of secreting cell wall around them. So let's say we have this bacteria, so they will create one, they already have a peptidoglycan cell wall, they will create one more cell wall around it, then second cell wall, third cell wall. So they have a very thick four layered wall around them. Okay, this what secretion of so many walls, how does it help them during a stressed out condition? The reason is that the bacteria will be living alive inside this. It's like cocooned inside a thick layer, so it's less prone to any injury or desiccation or things like that. The bacteria will lay dormant inside it. Make sense? It will lay dormant inside this cell wall, these different layers. Okay, and like radiation cannot easily pass through it, right? When you have such a four layers around it, it's not easy to get to the actual bacterial cell, right? Even let's say if they are in a severe place where they have so much radiation exposure, right? And then so they help them survive through these stressed out conditions, extreme heat, desiccation. And when other ability of the endospores is that because the bacteria is laying dormant inside, okay, it's not dead, it's laying dormant, it's like having a nap, think like that. Okay, so what happens is when the favorable conditions return, then the bacteria will come out of its dormancy. As soon as you give it some water, some moisture, it will start, the endospore will start to germinate. Okay, and when it's germinate, it can produce a normal bacterial cell. It can undergo binary fission and can produce a bacterial cell. So bacteria, what type of bacteria form endospores? Like I said, the two species are Bacillus and Clostridium. These are the bacteria that can cause, Clostridium can cause botulism. Okay, this one can cause anthrax and botulism, and this one can cause tetanus. Okay, so all these bacteria, all these, not all these, but these two bacterial genera are capable of producing endospores. So some of the internal membranes, like we said, they invaginate inside the bacteria and they can mark the regions for photosynthesis or for respiration. Because it's a prokaryotic cell, so there is no special organelle like a chloroplast or a mitochondria. But still, like I said, cyanobacteria, if you are a cyanobacteria, then you are going to perform photosynthesis. If you are, of course, uh, they will also perform respiration, right? And other, every other bacterial type is going to perform respiration too. But because they don't have special organelles, they will just, some certain areas inside the bacterial cell will be designated for that. Okay, the nucleoid region, like I said, is going to contain the single circular chromosome. It can also contain plasmids. Okay, those little hula hoops, remember? They are small, independently replicating DNA. So when we say they are independently replicating, means when the bacterial chromosome is dividing, they are not going to participate in that replication. They'll do their own thing. Okay, they are independent. They are non-essential, they are not really needed, these plasmids, but they provide, like I said, a selective advantage like antibiotic resistance. So that if that bacteria has this plasmid, of course they are going to have that gene that is going to give them that selective advantage of, you know, saving themselves from the antibiotics. Make sense? Now ribosomes are present in them, and you know why we need ribosomes? Because the function of the ribosomes is to carry out the protein synthesis. Okay, so they uh, are, the ribosomes of the prokaryotes are definitely smaller than that of the eukaryotes. They are not the same size. So ribosomes of bacteria have a size of 70s, whereas the eukaryotes have 80s ribosomes. Okay, so they are smaller than the eukaryotic, but their function is still the same as them, meaning production of proteins and RNA content. Oh, their RNA content is also slightly different. Now, you know, we have different classification schemes which we relied on staining characteristics and other phenotypes, whether they are motile, whether they are non-motile, whether they are photosynthetic or non-photosynthetic. They can be unicellular, they can form colonies, they can be filamentous. Okay, they can form spores or they can divide by binary fission. They can be important in terms of causing diseases to humans, and it's not always true. They are always, they can be beneficial also to us. I know it's hard to imagine that, because we think if we are talking about bacteria, oh, it's going to cause us a disease, right? But that's not always true. They can be beneficial also to us, you know, like the lactobacillus. You wouldn't have your yogurt if it was not lactobacillus. Okay, okay, so some of the molecular approaches to classification are, they amino acid sequences of the main proteins, their percentage of the guanine and the cytosine ratio. So these are the different ways that we have classified bacteria. Okay, the by the amino acid sequence, okay, of some proteins, and the guanine to cytosine ratio. Based on that also we have classified some bacteria, okay, like the gram positive bacteria. And then you have nucleic acid hybridization, meaning mixing of single stranded DNA from two species. You will find out closely related species which ones have more base pairing, and also from the genome sequencing, okay, gene and the RNA sequencing. When we say that sequencing, so these things, and especially rRNA, you know what is rRNA? The ribosomal RNA, and also whole genome sequencing. So these are some of the approaches by which we have been able to classify the bacteria, okay. And based on these molecular data, there are several groupings that have been proposed, and they are found in the Burgess Manual of Systematic Bacteriology, second edition. The first edition was based on the morphological characteristics, right? But this second edition of the Burgess Manual talks about these uh, sequencing, large scale sequencing of the samples, and that has determined the incredible amount of diversity, okay. And it also gives us information that majority of these bacteria have so far not even been studied because we couldn't really culture them, okay. So in labs, you see the amount of bacteria that you grow, you think, oh, it's a lot of bacteria we are culturing, you know, but that's not true in reality because you have billions and trillions of these little creatures, okay. I'll end it at this for a little did because we couldn't really culture them, okay. So in labs, you see the amount of bacteria that you grow, you think, oh, it's a lot of bacteria we are culturing, you know, but that's not true in reality because you have billions and trillions of these little creatures, okay. I'll end it at this for a little talking about the bacterial genetics or prokaryotic genetics. So we know that they can reproduce asexually mainly, okay, but they do have mechanisms for gene transfers, and they are called horizontal gene transfer. And like I said earlier, I mentioned that there are three types of horizontal gene transfers are transformation, conjugation, and transduction, okay. So these three. So what we mean by gene transfer is that it's not like there is a male and a female gamete and, like, you know, crossing over is happening like it happens in meiosis that you guys are aware of, and that's how the genes are being swapped. Genes are not here really like swapped like as if it does in eukaryotes, but here there are certain genes from bacteria X to bacteria Y. Okay, so think like that. Means bacteria S is a different individual, right? Bacteria Y is a different individual, right? So now what is happening here is that there is little bit transfer of the gene from one cell to another. Let's say this is bacteria X and this is bacteria Y, right? Now, obviously, each individual has a different set of genes, right? So, when what happens is in conjugation, what happens is that a sex pilus is formed. This is called a sex pilus. So, now bacteria X is like your donor cell. It will form a pilus which will attach to the bacteria Y, and now bacteria Y is your recipient. It will receive that DNA. So this DNA is now going to travel through the pilus, okay, and it already has its own, but it's going to get some of the bacterial chromosome. Make sense? Like this, like that, you can say. So it's not actually, like I said, such an advanced mechanism like meiosis and crossing over. You guys must be aware of that, right? It's just simple transferring of few genes, and they'll attach. Make sense? So this is called conjugation, and this is accomplished through the pilus. It is called sex pilus also. You will can see it like that also in some books. Okay, so conjugation is mediated by these plasmids. Okay, so they will, plasmids, they are the plasmids. You have a bacterial chromosome, they will, you know transfer through this. And then transduction is also a kind of gene transfer. And what happens is, here there will be a virus called a bacteriophage. Okay, this is a virus. This virus attaches to the bacteria. Okay, and when it attaches to the bacteria, it now this bacteria has its own DNA or chromosome. Virus has its own. Okay, so this is a virus called bacteriophage. Have you heard of it? Phage in short. Phage means something that will eat. Bacteriophage means a virus that eats the phage. Make sense? So it's a phage, and what it does is, this is, let's say, bacteria called E. coli. It will be a specific bacteria for a specific phage. Virus will come and attach to the surface of the bacteria. It will inject its DNA. Okay, so this is the tail of the bacteria, of the virus, I'm sorry, and this is the head. Okay, this is the head and this is the tail, in short, just saying that. It has this DNA will be injected here, and virus has a unique way to uh, reproduce itself. Virus is actually considered a hijacker. So once it hijacks this bacteria, which is its host now, it's going to tell the virus that, hey, stop making your own type, make my type. So what happens is, the bacteria has to listen to it. Okay, it will stop doing what it is doing, because this bacteria is going to take over and it's going to reproduce many copies of the virus, not of the bacteria. Make sense? And but there is a little bit of bacterial chromosome there too. You know what I mean? It's not going to just like fade, but that's how, what I mean. The gene transfer has occurred now. Okay, from the bacteria gene, you got intermixed with the virus DNA. Make sense? So this is a gene transfer, right? You have the gene of both the bacteria. Make sense? Now transformation is slightly different. Transformation occurs because of the environment that the bacteria is in. Okay, so what happens in transformation? So this is conjugation. You see, there is a pilus being formed from one bacteria to another, and that bacteria has gotten that plasmid. So this plasmid is encoding for fertility factor. F factor means a fertility factor. So just like antibiotic resistance gene, a plasmid can have a fertility gene. Okay, so F plus cells contain that plasmid, F minus cells do not contain that plasmid. So let's say this is F plus, this is whatever I had called X, let's say it is F plus, and it has this plasmid, or this is F minus, it does not have this, so it gets into here. Make sense? Like that. So F minus cells get that gene, fertility gene, and then they give it to the recipient cell, that also becomes F positive. So F, this is another way that is represented in the picture. Okay, the F plasmid will bind to the site, to the interior of the F plus cell near the pilus, now called a conjugation bridge or a pilus. So F plasmid is replicated, and then one parental strand of the plasmid DNA is displaced. The displaced strand is transferred to the recipient cell. Okay, a complementary strand is added, creating a new stable F plasmid. After successful transfer, the recipient cell, after that, this is your recipient cell, this cell will also become F plus. Make sense? Make sense? To begin with, it was F minus. Okay, so uh, this is basically the story. Then you have HFR cells also, which means um, high frequency of recombination. Okay, here the plasmid is replicated every time the host divides. And the plasmid can be integrated, and then it can be excised also easily. I'm kind of cutting it a little bit short here. You don't need to know so much detail. But then same way, just like you have H factor, you have the R factor or resistant plasmid also. These are the ones that are going to carry the antibiotic resistant genes. They have they have those genes that will code for resistance to antibiotics and bacteria. Okay, so it acquires genes through transposable elements, its selective advantage in the presence of antibiotics, and that's how we are able to produce resistant strains of Staphylococcus aureus. And there are some virulent plasmids, and they contain genes for pathogenic traits. You know what do we mean by pathogenic traits? The ones that end up causing disease. Okay, so that those are virulent plasmids, and that has happened in the a strain of E. coli called O157H7. So normally E. coli is not so infectious, but this strain has gotten this virulent plasmid, and it has, arose this way. So this is called harmful strain. This is the harmful one that can cause disease. You might have seen E. coli O157 causing diseases like diarrhea and upset stomach and things like that. Okay, we have had cases in past for this disease where it caused like in lettuce or spinach, the harmful E. coli O157 was found, and it was causing sickness in people. Okay. Now, generalized transduction, there are two ways, like I already explained. This is generalized, then you can have specialized also. So, generalized occurs because of the lytic phage of the lytic cycle of the phage. What is the lytic cycle? In lytic cycle, what happens is that when the E. coli is infected by the virus, then the virus will come out of the cell by the lysis of the host. Okay, so now the virus, assembly will take place. They will form this capsid around them, and then they will be released by the destruction, lysis of the E. coli. Okay, that's how generalized transduction is called. But specialized is caused by the lysogenic cycle of the phage. What is lysogeny? In lysogeny, what happens? It's the similar way that they will infect the bacteria, but instead of lysis, what will happen is that the virus is going to undergo like this. So let's see, Now the bacterial, this is the bacterial chromosome, right? What it will do is, in this case, it will sneak inside the viral DNA is going to sneak inside the bacterial DNA and will become part of it. Okay, so in this particular process, this is called a prophage. The bacteria is existing with it has integrated itself with the bacterial chromosome, right? And now bacteria will think that, okay, I have to divide. Bacteria will normally divide, right? But when it is dividing, it is also replicating the viral DNA with it. Okay, so it's pretty smart like that. The virus phage is very smart. So anytime it's creating its copy, it is also creating the back viral or the phage DNA also with it. Make sense? So this is called specialized transduction, and the prophage encodes the functions necessary to excise itself and undergo lytic growth. So if the what happens is, it will, after multiple rounds of multiplication, what will happen, this phage will excise itself from here. Okay, and once it excise means what will it do is, it is going to form, you know assemble itself like that. Make sense? Once the many viral copies are formed, it is going to excise itself. Okay, and at this point, lysis will occur. It will break down and the cell wall of the bacteria will break down, and this will come out like that. Make sense? So earlier, this is called lysogeny, this is called lytic cycle. So they can undergo these viruses can undergo two types of um, cycles. Lytic means it will cause the lysis, means it will burst open the bacterial cell wall in order for them to release when they are ready to get released. But in this case, they are going to lysogeny is longer. Why? Because they are going to integrate themselves with the bacterial DNA, and then when it's time for them, after a certain time, they can remain in this situation for years, without causing any symptom. Is like in AIDS and all, people don't even know until like 10 years or so that we have AIDS, because this is what the virus is doing. It's integrated with the normal host DNA, and it's not causing any infection, it's just replicating. But then when it wants to release itself, it's going to cause the lysis, and that's when we come to know all those symptoms. You know what I mean? So prophage. Now what is transformation? I stopped there, I didn't explain it earlier, so this is why I was waiting for. So transformation is that the DNA fragments are released from a damaged cell, and they are just directly taken up by the live cell, you know and so what happens is, the new DNA is now part of the live cell's genome by recombination. Okay, it's just like that. Like, there are two ways you can do. One is natural and one is artificial. So naturally, if they can't go, it can be done in lab. So how does this happen? So transformation. So let's say we have like we can do it in we have R type bacteria and S type. S type is smooth, and this is the one that is pathogenic. Pathogenic means it is going to cause disease, and this rough is non-pathogenic. Okay, got it? So R type, let's say you killed this R type bacteria. This was heat killed, you know. Heat killed means you provide, give them heat and it killed. So, but I'm sorry, the other, this one, S type is heat killed. So this was heat killed. So then when they both are mix R plus S in a tube, they are together, right? So what happened is this is killed R type, but its DNA is still there. You know what I mean? And when you injected this in a mice, you found, this is my silly mouse, you injected it in a mice, right? When you, this concoction, heat killed HK, let me write it like that, HKS, okay, heat killed. So what happened is, you would think that because this is pathogenic and this is heat heat killed, you know what I mean, then it should not cause the mouse to die, right? Mice should not die, but the mouse dies. R is living, but what happened is it took away the DNA into itself and it became pathogenic. So this, what does this tell you? That the transformation of the R type occurred, right? It acquired the gene because they are both in the same environment, in the same petri dish, same test tube, however you want to say. R type was able to acquire the DNA of the S type and itself became pathogenic. Remember, begin with it was non-pathogenic, but now it became pathogenic. Okay, so that's how, this is what we mean by transformation. The other type became, it got transformed into pathogenic type. Make sense? So artificial transfer, normally, naturally also this happens in many bacteria, but if it doesn't happen, then you can artificially also induce transformation. Got it? So this is critical for gene cloning and DNA manipulation. Now, looking at the metabolism of these prokaryotes, you have these um, four types at least. Photoautotrophs. What are photoautotrophs? Photo means light, autotrophs means ones who can make their own food. So in that category, you know, we have the cyanobacteria because they are going to use the sunlight energy. Okay, so photo is coming from there and they are going to make, convert the inorganic carbon oxide to carbon dioxide to organic carbon, meaning they are going to, you know, as a result of photosynthesis, we make organic molecules like what? Like carbohydrates, right? And oxygen is produced. Now, there is another type of bacteria called sulfur bacteria. They will use hydrogen sulfide as the electron donor because in the absence of oxygen, which happened, you know, before cyanobacteria came on the earth, right? Before, there were some purple sulfur bacteria and they don't have, because there's no oxygen in the environment. So as a, you know, in aerobic respiration you need oxygen as a final electron acceptor, right? Because no oxygen was available, so they just use hydrogen sulfide because that's what was present in the deep hydrothermal vents, right, in the deep in the sea. So that's another way. And then you have some uh, chemolitho autotrophs. What does that mean? Means they are going, chemo represents chemicals, right? And they will produce energy from oxidizing the inorganic substances. Okay, and yet another type is the nitrifiers. What are nitrifying bacteria or denitrifying or nitrifying bacteria? They will oxidize ammonia or nitrate. If they are nitrifying bacteria, they will oxidize ammonia. If they are denitrifying, they will denitrify the nitrate. Okay, and usually these type of bacteria are present in the root nodules of many legumes. Okay, archaeal species also use simple form of photosynthesis. So chemo heterotrophs are the ones that will use both carbon atoms and energy from the organic molecules, and most prokaryotes are like that. And photoheterotrophs are, they are going to, just you had photoautotrophs, this is photoheterotrophs. This means what? They are going to use sunlight, but they cannot make their own food, they don't have capabilities, so they are not going to undergo photosynthesis, but they will use light as the source of energy, but will obtain their organic carbon made by other organisms, meaning the ones that was made by cyanobacteria, but they can use it, right? Just like we eat, like the other day I explained you, how do we survive? By eating the food that was made by the photosynthesizers, right? Purple and green, green no sulfur bacteria are also there, and then you have chemo heterotrophs, which will use carbon atoms and energy from the organic molecules. And then there are some decomposers and some are pathogens. So when we talk about pathogens, we know that in the early 20th century there were a lot of infectious diseases present, and 20% of the children were getting killed before the age of 5, even because we did not have much sanitation processes, hygiene, cleaning hands, desanitizing. These things people were not aware of. That's why we had those infectious disease, and it was very easy that people could easily contract those diseases and would die. But as sanitizers were evolved, and then later antibiotics came into being, the condition improved slowly. Now, tuberculosis is a disease that is caused by a very deadly bacteria called Mycobacterium tuberculosis, and it affects the respiratory system. Okay, it is kind of, this disease is second to AIDS, as a cause of death. 10 million people in 2017 were contracted, they contracted TB, and 1.6 million died from that disease. It can, because it's a respiratory disease, it can spread very easily from one person to another. Then you have diseases like peptic ulcers. So these diseases are uh, generated by Helicobacter pylori, which is found in such a acidic environment inside our stomach, so it is so resistant to the acid that is produced by the stomach. They, and that's why you have those ulcers. Then dental caries is like the plaque, dental plaque, which is like the bacterial biofilm, those that ends up causing the tooth decay. People, extreme cases, people can have periodontitis, they can lose the bone, and all the teeth can fall out. Then you have Streptococcus, Soprinus, and Streptococcus mutants, they all ferment sugars to lactic acid and tooth enamel degeneration, leaving tooth vulnerable to infection. So these are some of the important human bacterial diseases. As you can see, anthrax is there, Bacillus anthracis, and then botulism is food poisoning is caused by Clostridium botulinum. All these things. Cholera is caused by Vibrio cholera, dental caries, diphtheria, Corynebacterium diphtheriae, gonorrhea, Neisseria gonorrhea, and then leprosy caused by Mycobacterium leprae. So these are very, very important diseases